right, in this session, we're going to learn about how to interpret pH versus volume titration curves. You can find more information about this in OpenStax Chemistry, section 14.7, and I really strongly recommend this linked Libre text article on acid-base titrations. The Libre text is more thorough than OpenStax here, so I really encourage you to check that out. First question is, what is a titration? If we're doing this in the lab, you can imagine that maybe it looks something like this. You'd start with an analyte, a solution that you want to know something about, like you want to know its concentration. And you slowly add the titrant, another solution that you do know something about, in order to learn more about the analyte. In our case, we're mostly looking at acid-base titrations. So we're going to have some reaction of an acid, say, so maybe this is something like HCl, and then maybe our titrant is NaOH, and we know that if we mix those, we're going to make water and NaCl. And so here we're controlling the amount of NaOH that we add, so that we can learn more about the HCl solution. And so if we were to measure the pH of that mixture over the whole range of NaOH that we add, we could see that the pH would change like this. So in this graph, you see we have pH as a function of how much NaOH we've added to a strong acid like HCl. So basically, this is a sort of series of stoichiometry problems. At the beginning of the titration, we could calculate the pH based solely on the HCl amount. So HCl is the source of H3O+, and we could calculate the pH based on its concentration. Then when we start adding the NaOH, you can see that the pH changes, and in this before equivalence point region, what's going on here is that the NaOH, the added thing, is the limiting reactant. And so we can calculate the pH based on how much of the HCl is neutralized. After the equivalence point, the HCl is the limiting reactant. And so that means that at the equivalence point, a really important point in a titration curve, at the equivalence point, the amount of NaOH added equals the HCl that we had initially. Or in other words, there's no limiting reactant. They're in their stoichiometric equivalence. So there are three really important things that we can learn from titration curves. We can learn whether the substance is a strong or weak acid or base. We can learn what the initial concentration of the acid or base was, and we can figure out the pKa or pKb of a weak acid or a base. So the first is we can tell from the titration curve whether we're looking at a strong or weak acid or base as our analyte. So here we have superimpose the titration curve of a strong acid and a weak acid. These two have different shapes. So we can see that before the equivalence point, the strong acid is all concave up, and then we see that the weak acid has an inflection point. These two have the same equivalence point in terms of volume. So here we can see that for the weak acid, so we're talking about the weak acid's got an inflection point right there, whereas the strong acid doesn't. And we see that the equivalence point has the same volume for both of them, although the equivalence points have different pHs. So we see that the equivalence point for the strong acid, the pH is about 7, but for the weak acid, the equivalence point here is about 9. For a weak acid, the pH equals the pKa at the half equivalence point. So here the equivalence point is about 25 milliliters. So the half equivalence point, when half of that's been added, we're at about 12 milliliters added. And we see that that's the same as that inflection point along the curve. So we see where the concavity changes. And so the pKa of this weak acid is about 5 because that's the pH at that half equivalence point. So what kind of titration is shown in this titration curve? Here we have the titration of a weak base with a strong acid. Okay, we know that it's a weak base because our pH starts basic. So when we have no added acid, we see that we have a pretty high pH. We also know that we have a weak base because we see here this inflection point where the concavity changes. We know that it's a strong acid because in general, our titrant, the thing we add, is always either a strong acid or a strong base. If we titrate with a weak acid or base, then our math gets really complicated and we learn less from doing that. So this is going to be whatever the added thing is always going to be strong. So here's another one. This plot represents the titration of a weak acid with a strong base. So again, we know that it's a weak acid because we start acidic and because we have this inflection point where the curve flattens out, right in here. Okay, so the second thing that we can learn from a titration curve is what was the initial concentration of the acid in the base in the sample. And we can do that because we know, we can find on our titration curve where the equivalence point is. So if we look at this curve, what volume of titrant needs to be added to reach the equivalence point? So the equivalence point is where our graph is almost vertical. So we see that here the pH changes very quickly with a little bit of added titrant. And we find that when we have that inflection point, that represents the equivalence point when the volume or the moles of titrant equals the moles of analyte initially. And so we're looking for the volume where that happens. So here, the volume of titrant is 25 milliliters to reach the equivalence point. And what that means is that the moles of titrant added exactly equal the moles of analyte initially. We can then use that to calculate the initial concentration of the acid solution. So if you think about what was the concentration of the analyte when we poured it into our titration apparatus. So in this problem we have a 30 milliliter aliquot of acid solution was titrated. So if we picture what that looked like, we had our Erlenmeyer flask and we put something like 30 milliliters of acid in there. Aliquot just means a portion. We titrated that with 0.123 molar NaOH, and our question is, what was the initial concentration of the acid? Okay, so we want to know the moles per liter of the acid solution. Do we know either of those? So in order to find the moles of acid, we are going to use our titration curve. So we said that the moles of acid equal the moles of added NaOH at the equivalence point. We said that the volume of added NaOH at the equivalence point was 25 milliliters. So for the NaOH, so that we know that moles of NaOH is going to be 25 milliliters, or 25 times 10 to the minus third liters, times the concentration, 0.123 moles of NaOH per liter. And if we do that, we find out that the concentration of, or the moles of NaOH is 3.08 times 10 to the minus 3 moles. Okay, we said that is equal to the moles of acid that were initially in the analyte. Okay, and so then to find the molarity of that acid, we have the moles of acid, we need the volume. Here the volume is that initial volume, 30 milliliters. And so if we divide that number of moles of acid by the initial concentration of that acid solution, we find out that the concentration of the acid solution was 0.103 moles. So this ends up being a really useful measurement technique for figuring out what is the concentration of a solution when you don't know what the concentration was. The final thing that we can learn from a titration curve is what is the pKa or pKb of a weak acid or a base that we titrate. So all of the pKa's and the pKb's that we list in tables somewhere have all been measured using experiments. And this is one example of an experiment that we could use to make that kind of measurement. Okay, so what is the pKa of this weak acid? There's a couple of different ways that we could analyze this curve. One of them, so for starters, we said that the pKa equals the pH at the half equivalence point. On this curve we looked before, we said that the volume of the equivalence point is 25 milliliters. So that means that the half equivalence point is 12.5 milliliters. And then we can look for the pH at that point and find out that the pKa here is about 5. Another way of looking at that is that the pKa is the pH at this inflection point where the curve changes from being concave down to being concave up. Or in other words, where the slope equals 0. If we have a titration of a base, a weak base with a strong acid, then instead of giving, then we can find the pKb of the base. So can you work out the pKb of this base? Okay, so we can find the half equivalence point. We see here that the equivalence point is 50 milliliters, so the half equivalence point is 25. That's where that inflection point is. And we see that the pH there is, call it 9.25. Now, that 9.25, because this is pH, we said that that equals pKa at the half equivalence point. Equals 9.25. So that means that the pKa of the acid form is 9.25. To find the pKb, we need to subtract this from 14, pKw. So if we do that, we find out that that means that the pKb is 4.75. And so again, we need to subtract from 14 because this axis is pH. So pH is going to give us pKa. If we were plotting the pOH, then it would give us pKb. But that would be a different experiment that we basically never do. We can see that for different acids or bases, that we can see how the titration curve changes with the different pKa's. And we can also look at how the pH at the equivalence point changes. So for a strong acid and a strong base titration, the pH at the equivalence point is always 7, because when we titrate a strong acid and a strong base, when they exactly neutralize each other, we always get neutral water. We see that the pH for weak acids and bases is at the equivalence point is higher. So we have basic solutions at the equivalence point. And we see that for weaker acids, so higher pKa's, that the pH at that equivalence point steadily rises. So if the pKa was 4, our equivalence point's about there. For pKa of 7, equivalence point's even higher. For pKa of 10, equivalence point's way up here. And the sort of opposite is true for bases. So for strong base and strong acid, again, the pH at the equivalence point is about 7. For pKb is 4, so again, a comparatively strong base, we see that shifts down to maybe here. The pH at the equivalence point for pKb equals 7 is even lower. And for pKb, for the weakest base, the pH is even lower. So we can use that to identify the relative strengths of different acids. So if there are 3 different acids that you titrated that had the pH at the equivalence point of these values, which of these is the strongest acid? The strongest acid is the one where the pH at the equivalence point is closest to 7, is the strongest. If we have a diprotic acid, we see that in the same way that we considered the sequential ionization, so that we did one ionization and then the next one, we can see that in our titration curve. Here we have actually a triprotic acid, where we have the first equivalence point, the second equivalence point, and the third equivalence point, and we can see the pH equals the pKa for each of the 3 values. For this acid, unknown acid, what are the 2 pKa values? This is a diprotic acid. We see that the 2 pKa values are 5 and 10. And the way we figure that out is the same way we did before, where we can look for the 2 equivalence points and then look for the 2 halfway points, or the 2 places where the slope of the curve equals 0. And we see that the pH at those half equivalence points is about 5 and about 10. Okay, so again, in this session, we learned how to interpret pH versus volume titration curves.